Hey, it's Horner, and this is how to make a protein or protein synthesis. This process has two steps, as you see, and in step one, we're going to do transcription. Remember, transcription has the C in it, so that comes before translation, which is step two. Transcription does happen in the nucleus, so you'll see here's the phospholipid bilayer. To the left is the nucleus, and to the right is the cytoplasm. So that is just the other part of the cell, and the nucleus would be inside. So very first thing that we can do is we can zoom into the DNA, and we'll kind of see what happens. So this is just a good overview. When I click the button, you'll notice the DNA unzips, and then messenger RNA comes in. And that is the process in general called transcription. That transcript then is sent to the cytoplasm where it's read on the ribosome, and we make the protein from the amino acids. So let's go ahead and take a look at this next part in a little bit more detail. So in this part we've got DNA and we have RNA. And so the DNA is double-stranded, RNA is single-stranded. This uses deoxyribose nucleic acid, and this is ribose nucleic acid. So that's the difference between those two. Every gene has something called a promoter region and a protein coding region. And so what's going to happen here is in this promoter region, we're going to attach something called an RNA polymerase. So we'll go ahead, and that is the RNA polymerase. So that's what that looks like, and you've seen that in some other notes too. To begin the transcription, that RNA polymerase attaches to that promoter region. And inside the promoter region, then, the gene's DNA strands are unwound. So at this point, what we're going to do is make an RNA transcript. So we're going to read inside that region. Kind of what happens is it basically takes the code from the DNA and makes the RNA code. But remember, instead of going through and pairing T and A, we're going to pair T and U. So U will replace that as we go through instead of using the other. So this one, we've already built the transcript. So you'll notice wherever we have a T on the DNA, we do have an A on the RNA. But anywhere where you have an A on the DNA strand, we have to put that U. And so it would be the phosphate, it would be that ribose sugar, and then it would be the nucleotide is what you're looking at there. So we're going to go to the next part, and then the RNA polymerase basically just forms bonds or links between those nucleotides to create that transcript. So now we're going to bind them together, and you'll see that that then pushes out. And now we are in the protein coding region. So this is that RNA transcript. And you'll notice that it just moves right along, both the DNA and the RNA. The DNA is unzipped inside of the polymerase. And that RNA polymerase is really just an enzyme that puts together all of these chains, or this big chain of RNA. In all those little units. So now you kind of see how that works. To finish the transcription, you'll notice that this mRNA then gets cut and it moves away from the RNA polymerase, and the DNA then reconnects itself. That messenger RNA then moves out of the nucleus. And so we're going to send it to the cytoplasm. So you're going to see how it kind of moves through the nucleus, and then it goes right through this little guy here, And that allows, so that's called a nuclear pore, and that allows it to move right through. So now we have completed transcription. Next thing we're going to do is let's take a look at translation. So in translation, remember that happens in the cytoplasm. So we're going to zoom in on the on our messenger RNA, and we're going to take and send this messenger RNA to a ribosome, and it's got two parts, and it closes down on top of that message that we have from the messenger RNA. They want to show you here how that ribosome translates the sequence of nucleotides in messenger RNA into amino acids. So we're going to click on that. And remember, every 3 is called a codon. So you have 3 nucleotides together. So there's codon 1, codon 2, codon 3, codon 4, and it just keeps going. So at this point, we would code for the amino acids, and it shows a little bit how to use that codon wheel. So almost all of these will start with AUG. Remember, that codes for methionine. So we're going to do A, and then we're going to do U, just like in your wheel, and then we're going to select G. And at that point, we get methionine, which is a start codon. And that means we're going to start moving through and building this amino acid chain to make up a protein. So the second one here is G, and we're going to do C, and then we're going to do A, and that codes for alanine. So we're going to submit that guy, and now we have codon 2 is alanine, and then codon 3. This one is CUG. So we're going to do C on the inside, then we're going to do U, and then we're going to do G, and when we do that, we see that we get leucine, and we'll submit that. Now that we've done those, we're going to go on to number 4, and this one is U, and then C, and that's phenylalanine, so we're going to submit that one, and then we'll go on, and now we have all of the amino acids that we want for these 4 codons. The transferrin RNA then comes down and it links to the codons that match it, and this one is methionine. So it will kind of come down. You'll see this is the amino acid, and the tRNA here will then come down and it'll see this code, and then that methionine then is coded right from the tRNA. So you'll see the letters match up. This is what we read on the mRNA, and then this is what this is called an anticodon, and it is on the tRNA. So codon is on the messenger RNA, and the anticodon is on the tRNA, and so that one has methionine on it. So now we are set and we're ready to go, and we see that this thing is going to carry that amino acid, methionine, so everything's been read, and then the next codon is CGA, and so it'll have us go through and pair, and then you'll have another one, and you just keep going. So these T and RNA just keep coming in, and we keep adding more and more amino acids to the chain, and that pushes out that big strand of amino acids, and that's what makes up the protein. So when we get to the end, there's a stop codon. So right here, we see the letters are going to be for a stop codon, and that just means that when it comes in, it is called a release factor. So that's the release factor. Now we've pushed that protein out, and now that protein is going to kind of take off and go do its job in the in the cell. So this amino acid string that we just made folds into a protein, and this is called an ADA enzyme. So it's going to do its thing, and it will fold around, and now this protein is ready to do its job. So we can also unfold it so you can see it back and forth. But basically, that's what it does and how the whole process works.